We're going to talk about the nervous system, which is one of two communication systems that tie together, link together, allow communication to occur between the different organs and organ systems in the body. All right. The one that we're not talking about, we've made references to it throughout the course, is the endocrine system. All right. And the endocrine system, just by way of comparison, is a diffuse system. It's a tissue releases molecules into the blood circulation, right? And those molecules circulate, and because of the nature of that system, they can have multiple targets and have very widespread action. All right, when you think about adrenaline, for example, all right, it has all kinds of effects on your heart and on your respiratory rate and on your metabolic processes and so on, blood vascular system throughout the body. So you have very widespread kind of an effect. But those effects are somewhat slower, all right, and the typical kinds of events that are controlled, apart from things like the actions of adrenaline, are more slow, long-term, sometimes cyclic kinds of events by the endocrine system. All right? We're going to look at the other communication system, which works in a very different way. First of all, it's a hardwire system. There's a physical connection point to point from the control center to the effector, the tissue that you want to have an action affected on, all right, which would be muscle or gland, or to conduct a message to an adjacent nerve cell. Okay, so it's a hardwire system. It's a point-to-point system. All right, it is based on what we'll call an electrochemical impulse, which we won't go into in this course, but you'll learn about it in physiology. And it is simply the movement of ions across the cell membrane in a very sequential manner that is carried down the length of these nerve cells and leads from a signal being carried from one point to another. And we'll sort of leave it at that for right now. All right. I guess one could add that as we talked a lot about your kidneys and their role in terms of regulating the ion concentration of the body fluids, if the ion concentration of the body fluids becomes too out of balance, it will affect nerve tissues, all right? And sometimes you see the effects of that when people run a marathon and they've been exerting themselves for hours and perspiring and losing electrolytes, and they start, as they get to the end of that race, sometimes they actually start to exhibit signs of neurological failure. All right, and there's a couple of famous ones where the runner enters into the stadium and runs the wrong way around the stadium in the final lap and so forth, things like that, where they're delirious and they're wandering around and so forth. All right, so and that's because of this ionic imbalance, which is required for nerve cell functions. Okay, so when we look at the components then of this system, which again we look at it as a system for coordinating rapid, short-term, repetitive kinds of activities, all right? That's where your nervous system's going to come in. We see the three sort of components, and we're going to come back to these again and again tonight, and that is a sensory component. This is the incoming or afferent, all right, component of the nervous system, all right? It's going to pick up stimuli, whether they're physical stimuli in terms of mechanical changes, stretch, touch, vibrations, those kinds of things, or chemical changes in the body relating to concentration of oxygen or CO2 or pH changes, pain as a warning of damaged tissues, and your other senses, chemoreceptors for olfactory, for taste, photoreceptors, all right, stretch receptors of different types. So all of these different sensory systems are part of the incoming or afferent information coming into the nervous system, all right? That's going to carry its information into the processing center, all right, where integration and processing occurs, where evaluation of that sensory input occurs, and hopefully some kind of an appropriate response is mediated, all right? And then finally, an efferent signal is sent out to the effector, all right? So we bring about some kind of a result. A response is dictated by the central nervous system, all right, to the effectors, and again, the effector tissues are going to be muscle, skeletal muscle, of course, in the carcass, smooth or cardiac muscle, all right, in the visceral cavity. All right. Any smooth muscle out here in the carcass? Your blood vessels, all right, and your little erector pili to make your hairs stand up. So there is, and so we want to remember that. We don't want to just think of the carcass as all skeletal muscle, all right, but there is vascular tissue out there. You're going to have vasomotor activity. We've got to control that as well, and we'll come back to that a little bit later, all right. So effectors are muscle, gland, all right, the secretion of glands. Think about the pancreas as a result of, you're eating food. Think about your adrenal gland secreting its product as a result of some stimulus, right? You were doing your text messaging, and you looked up because you were driving, and all of a sudden you look up and see brake lights, and oops, I shouldn't have been doing that. You hit the brakes, and you feel your heart pounding, and so forth. And those were nerves that acted as sensory, picked up that signal, sent it down to the adrenal gland, and the adrenal medulla secretes, adrenaline or epinephrine as a result, right? So you have nerves acting on a gland. All right. You also have nerves acting on other nerves, right? Throughout the brain, you have nerves synapsing with other nerves. And so we can think about sometimes output involves nerve-to-nerve signaling before it goes on to the effector. And we'll say more about that. Okay. So from a structural standpoint, as we look at sort of the anatomical arrangement of the nervous system, we can subdivide it into the central nervous system, which includes the brain and the spinal cord. All right. I unfortunately am not going to talk about the brain anatomy in this course. If we had more time, we probably would do that. That'd be probably the next thing we do. But we're going to run out of semester before we run out of material. But if we look at the brain, we can think of its three major components. All right. The cerebrum, which is the largest part of the brain. This is where processing goes on. This is where memory goes on. This is where your thinking takes place. Your ability to imagine things, where you take that sensory input and you create images of that and things that you can process and contemplate. All right, that's all happening in your cerebrum. All right, this is where you initiate activities from, as well as interpret the world. All right, this smaller portion of the brain back here is the cerebellum. All right, and its purpose is coordination and what we call motor memory. All right, motor memory, of course, is just that idea of when you bend over to tie your shoe, you don't have to go through the rabbit went under the bush and around the tree and over the stump or whatever it is that you memorize to tie your shoes. You just do it, right? It's the golfer just doing it naturally, swinging. That's motor memory, all right? And then coordination is the idea of seeing that baseball coming to you and knowing when to put your hand out and to grasp that ball, right? That's coordinating your visual sensory input with some motor output. So that's your cerebellum. And the brain stem, that's doing all your involuntary control systems. So where the cerebrum says, I want to lift something up, and you engage those skeletal muscles into play, all right, the brain stem is controlling vasomotor activity. It's changing the heart rate. It's increasing blood respiratory rate. It's changing body temperature. All right. So all of those things that are controlled unconsciously. Right. All those visceral systems that need to be controlled. All right. And then the other component of the central nervous system is the spinal cord itself, and we'll be looking at the spinal cord more in detail in a bit. All right. So together, those are the central nervous system. And the afferent fibers come into the central nervous system, and the efferent fibers leave from the central nervous system. All right? And those afferent, approaching, and efferent, exiting fibers make up the peripheral nervous system. All right? And they include then nerves that emerge directly from the brain, called cranial nerves, of which there are 12 pairs. All right? And those nerves that emerge from the spinal cord itself, in between each of your vertebrae, all right, paired again, 31 pairs, and these nerves, again, they are sensory, afferent, bringing information into the central nervous system, all right, they are efferent, motor, all right, and most of these nerves, and we'll break it down later, most of these nerves are mixed nerves, which is to say that these nerves that emerge from the spinal cord, all of those nerves that emerge from the spinal cord, all of those are mixed nerves. They carry both motor and sensory information. They have afferent and efferent fibers. All right. Among the cranial nerves, some are exclusively motor, some are exclusively sensory, and some are mixed, and we'll identify those later. All right. So we'll break that down a little bit later on. All right. And I do want you to know those later on. So that's just going to be a little task of memorization. All right. The other part of the peripheral nervous system is what we call ganglia. And typically, when we think about the cell bodies of nerve cells, they almost always, or the vast majority of the cell bodies of nerve cells, are found in the central nervous system. They're found in the brain. They're found in the spinal cord. They constitute what you know of as gray matter. All right? And it is gray matter because it is that part of the nervous system where the nuclei of the nerve cells are found. All right? And most of those are in the central nervous system. However, there are a few instances in the body where the nerve cell bodies are found outside of the central nervous system and where synapses occur with those nerve cells. And these are called ganglia. All right? So ganglia are little clusters of nerve cell bodies found outside of the central nervous system, found out in the periphery, okay? And we'll make note of a number of these ganglia, all right? And they're pretty important and easy to sort of appreciate, and we'll come back to those. All right. If we shift from an anatomical or structural organization of the nervous system to one that puts the emphasis on the function, all right, we can take the nervous system and subdivide it into the sensory side and the motor side. And I've confounded the diagram here by adding a bunch of arrows, thinking that it was clever, and now I sort of regret it because it just adds confusion. But what I'm trying to say is this. You have sensory carrying information into the central nervous system. You have motor carrying information out of the central nervous system. Let's look at each of these, all right? From the sensory side, you have receptors that are picking up information. Sometimes those receptors are the nerve cells themselves. Sometimes those nerve cells depend on secondary cells that are right at the very source, right at that site where the stimulus is being detected, all right? And those are sometimes called secondary receptors, all right? An example would be in your taste buds. You have cells that are sitting there on the surface of your tongue that are secondary receptors, and those in turn signal to the nerves, all right, to carry a signal relating to some taste experience, all right? Likewise, your photoreceptor cells, your rods and cones, those are secondary receptor cells that are then synapsing with nerve cells that form the optic nerve, all right? And so you sometimes have receptor cells, sometimes the stimulus is picked up directly by the nerve cell itself. We can take the sensory component and we can subdivide it into somatic sensory and visceral sensory. And we've talked about this before, right? I told you somatic sensory comes from the carcass, right? That is in the form of all kinds of different stimuli coming in from the surface, right? You can detect temperature, you can detect pressure, you can detect vibrations, you can detect pain, all right? A whole variety of stimuli, as well as the position of your joints in space, right? We call that proprioception. We talked about that before. All right, so you have somatic sensory then, carrying sensory information in from the carcass. And among those sensory inputs is pain, which is sharp pain, as I think I've told you before. All right. On the other side, and one final thing, you have a conscious awareness of all of those things, except the sensory that's coming from visceral components, all right, where sensory from the blood vessels, you don't know that the blood vessels out here are dilated or constricted. You don't need to know that, right? You don't need to be absolutely aware of that. But you have a great awareness of the inputs going in from the somatic tissues, from the carcass, all right? There's also visceral sensory, and there's again, you're detecting temperature, you're detecting pH, you're detecting stretch, you're detecting all of these things, but you do not have conscious awareness of any of them except pain. All right, except pain. So that means, you know, who ate dinner tonight? Where is that dinner right now? Can you tell me exactly where it is? Is it in the stomach? Is it in the duodenum? Is it in the ileum? Do you know where it is? No. All right, it's there, and it is stretching those organs, and it is causing them to respond appropriately, but you don't have a conscious awareness of it. However, if you had eaten something that was spoiled, all right, and it was initiating other kinds of upset there, you would have a sense of pain that you could be aware of. Okay, so the idea here then is to recognize that the sensory input from the carcass is a separate system from the sensory input from the viscera. Okay? And likewise, as you're going to see, when we go to the motor side, it's likewise separated. Okay? So let's go around to the motor output side then from a functional perspective. This is, again, efferent exiting information. This is carrying motor output. On the somatic side, it is carrying voluntary information to skeletal muscle, period. All right? That's it, right? Somatic motor goes to skeletal muscle, period. So all the other things, of course, in the body cavity, there is no skeletal muscle in the body cavity with the exception of the diaphragm, which sort of is simply a partition there, all right? But apart from that, there is no skeletal muscle. So all of the motor input going into your, or I should say motor output, going to the viscera is via the autonomic motor, right? It's involuntary, going to cardiac muscle, going to smooth muscle, going to glands, and going to ganglia, all right, as you're going to see. So we have, as you see in my figure here, I've done one little slightly clever thing here to show you symbolically. All right, motor input, or I'm sorry, sensory input here, one cell goes the distance. One cell, all right, apart from when you have a secondary receptor, but when we get to the nerve cell itself, one cell, all right? So that one cell that I stimulate here extends all the way up my arm, all right, all the way into the central nervous system, running the whole. That's a big cell. That's a long cell, all right? And of course, it's a bundle of them, but each one of them travels the full distance, all right? On the motor output side, the same is true of somatic motor. One cell out to that skeletal muscle, all right, in a bundle, so many the full distance, all right? When we talk about autonomic motor, however, all right, this involuntary motor component going out to control what? Your visceral organs in the body cavity and blood vessels and erector pili muscles out in the carcass, that's a two-cell system. One cell goes part way, the other cell goes the rest of the way. All right, it's a two-cell system. And if one cell goes part way, and then there's a second cell going the rest of the way, what does that tell you is in between those two? A ganglia. There's a synapse there and a little location where you have a bunch of synapses. That's a ganglia. All right, and so we'll look at those at that anatomy a little bit later. But now you have the basic idea. Somatic motor, a one-cell system. Autonomic motor, a two-cell system. Okay? Let's take a look at the functional unit here. That is the nerve cell itself, or neuron. All right? The neuron is the functional unit. You see a typical nerve cell here, although we're going to see that they have other configurations as well, but let's just use this one as our starting point. All right? This is the input end of the nerve cell. That is, this is the receptive end, and so when we sometimes say that this part of the nerve cell forms the receptive field. All right? Any stimulus anywhere on these fibers that are extending out of the cell body here, or stimulus directly on the cell body as well, all right, can initiate an impulse, all right, can initiate an electrochemical impulse. These fibers out here that are receptive are called dendrites, okay, and we'll see that those dendrites can vary in number, all right, and vary in their expanse, in their density, if you will. All right, and they will then transfer that information to the cell body. All right, the cell body, of course, is going to receive that information. We'll talk about what that sometimes means in terms of integrative activity for the moment, or later on we'll come back to that. All right. It also, because it is the cell body where the nucleus is and where most of the organelles of this cell are, it is responsible for maintaining the nerve cell, all right, the health of the nerve cell, for maintaining the nerve cell. And consequently, if you damage the cell body, that's it. All right. It cannot heal itself. All right. However, if you damage the processes leading into or out of the nerve cell, you can regenerate those. All right. And we'll say a little bit more about that as well. All right. Extending outward now, as we go carry the impulse away from the cell body, is a long fiber called the axon, and that is the conducting process. All right, that is where that electrochemical impulse is going to be transferred down the length of that fiber, eventually getting to the synaptic knobs at the end. And you remember those synaptic knobs when we talked about the motor end plate, where nerve cells synapse with muscle cells, as an example. We talked about those little synaptic terminals there, or synaptic knobs, all right, where neurotransmitters released. So the axon extends from the cell body and ends in a branched series of eventually synaptic knobs, all right, and sometimes that axon is wrapped in an insulating series of cells, all right, forming what's called a myelin sheath, all right? And the myelin sheath provides a means of increasing conduction rates, all right? The conduction rate of an axon that is unmyelinated is about 0.5 meter per second, all right? 0.5 meter in a second. There it is. Boom, all right? If you myelinate it, if you wrap it in these cells, all right, in this myelin sheath, it goes up to about 120 meters per second. That's a significant increase, isn't it? All right, right. It's almost 250 times increased conduction rate. All right, 120 meters per second. So that's the purpose of the myelin sheath, and we'll come back and look at that later. Nerve cells have a very high metabolic rate. All right, and they metabolize glucose, right? They metabolize glucose. So sort of different from when we talked about heart muscle and we talked about its penchant for metabolizing fat, right? Nerve cells aren't into metabolizing fat. They're all about metabolizing glucose, all right? They want a simple nutrient, all right? And they want a lot of it, right? Nerve cells have tremendous longevity, all right? The nerve cells that you had that formed during your embryonic development, they're still with you, and they will be with you to the end of your days, all right? Because most nerve cells, with the exception of a few nerve cells in the brain, were terminally differentiated early in life, and they will never divide again. All right, so you by the time you were born, or certainly by the time you completed your first year of life, you pretty much had all the nerve cells you will ever have. Okay, they will not divide again, and there's a good reason for that. The nervous system is a very complex system. It developed in a very systematic way, so that all the connections were formed sequentially. Right? If you have damage, you can't go back and say, I want to rebuild that, because the architecture is so complex, the system doesn't lend itself. It's a trade-off. You could have a really simple nervous system and be able to replace parts really easily, all right? Or a super complex system that's very highly integrated, which you give up the capacity to go in and trade out parts because the connections are so robust that you just can't go in there. The analogy I always use, I'm thinking about you, Michael, because I know you probably have some connection with the service, and I think about, there's something about the space shuttle, and it's this very sophisticated thing. And you know what? I'd like you to put a sunroof in there. Right? Can you do that? Can you put a sunlight on the top of the cockpit? There's all this wiring, all these details, and all the circuitry. Can you just go in and just do that?
They actually use charge 10,000.
Just for what?
Just for one line of computer code.
Just one line, yeah. So the idea you've got something that's incredibly complicated to go in there and make some kind of major alteration is impossible. Hence, you allow it to develop sequentially. Those cells become locked in. You've got to keep them because that architecture is so complex you're never going to change it again. And if you damage it, that's it. There is no way to go back and fix it. So it really speaks to the complexity of that neural architecture. All right. So given that basic sort of model of a nerve cell here, all right, we can go on to look at the more specific forms, because if I tell you that, hey, here's a sensory nerve cell, and you use that first picture that I gave you with the dendrites out there and then the cell body, where does that place the cell body? It puts the cell body out here in your fingertip if you use that nerve cell that I just gave you, right? Because you have the cell body with these little dendrites. All right, and that's not going to work, because where are the cell bodies? They're in or very near the central nervous system. So we need to come up with different forms of these, and these are the three basic structural forms of nerve cells. The first one is called a unipolar, sometimes called pseudounipolar. We don't need to go into that. We'll just call it unipolar neuron. And it has dendrites that give rise to a peripheral process. There's a cell body here with a little short single process, all right, hence a unipolar structure here, and then a central process. And what we're saying is this is distal and this is proximate to the central nervous system, all right? So the central process is close to the central nervous system. The peripheral process is out to the periphery. And if you think of this as a sensory neuron, all right, this is out on the surface of your skin. It gives rise to this long peripheral process. This is sitting just outside the central nervous system, the cell body, and then the last little half inch to get you into the central nervous system is the central process here, where then it synapses within the spinal cord. All right. So here you have then a nerve cell that has a very long axon. The peripheral process and the central process, PP and CP, as I abbreviate here, form the axon, and the cell body sits off to the side using this little single short process here. Okay? So again, this is typical of sensory nerve cells. We're going to see that the cell body sits in a region of the spinal, or just outside the spinal cord, in a little ganglion known as the dorsal root ganglion. All right, that's where the cell bodies sit, and I'll show you where that is in a moment. All right. A different form here, where you have dendrites and a peripheral process and a central process of sorts, but the cell body is now in between, is a bipolar neuron. These are found in the retina, and these are found in the olfactory epithelium, so they're pretty limited in their location. I won't say more about it, but you have dendrites with a process, an axon with a process, all right, going out to the terminals of the nerve cell. All right, and then you have the most common kind of motor neuron, all right, which is multipolar. And the key here is that you have a very big receptive field of dendrites. All right, a very elaborate receptive field of dendrites. And what does that enable? It means that when you have motor output, one cell acting on that motor neuron, like a democracy. Yes, contraction. Yes, yes, yes. Yes, 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 yes. All right. Field of dendrite. Signal will then be appropriate because it's taking into account all of that information that's coming in. So motor neurons have this multipolar architecture, as do a group of neurons called interneurons, which are found entirely within the central nervous system. And in both of those instances, motor neurons and interneurons, the cell bodies are in the central nervous system. Whereas in the sensory fibers, what did we just say? The cell bodies are just outside in the dorsal root ganglion. And let's take a look at that so we actually put a name on these things, all right? This is a cross-section of the spinal cord, Okay, and you can see the little butterfly-shaped area of gray matter here. All right, this region up here, the dorsal horn. This is the ventral horn. This is the lateral horn here. All right, so that's all gray matter. What makes it gray? Cell bodies. All right, so we're going to see cell bodies, and we just said that you have these multipolar neurons that form the interneuron, which is right here. There's an interneuron. It's entirely within the central nervous system. And multipolar neurons also form the motor component. Here it is, cell body in the ventral horn, extending outward through what's called the ventral root, okay, and going out to the effector, okay. On the sensory side, here's what kind of neuron is this? Unipolar. You can see there's the nerve cell body, and there's its little unipolar process here, and then you've got the peripheral portion and the central portion here. And this little broad thickening here, that is the dorsal root ganglion. All right? It is where the cell bodies of sensory nerve fibers. All right? Is it inside the central nervous system? No, it's just outside. All right? It's just this little widened part of this spinal nerve. All right, and so we have our sensory neuron, its cell body sitting out here in the dorsal root ganglion, or sometimes posterior root ganglion. We'll use the term dorsal root ganglion. All right, and now that the central process extends inward, and in the dorsal horn, it synapses. In this case, it synapses with the interneuron. So we begin to see the elements of a neural circuit, right? A sensory fiber picking up information, carrying it in its cell body in the dorsal root ganglion, synapsing here in the gray matter, all right, and then with or without the help of an interneuron, synapsing here with a motor neuron, all right, in the ventral horn, and that carrying out here, all right, eventually going to the effector. You're going to see that these two components, all right, this part of this bundle of nerve cells here that becomes the dorsal root, and this portion here, the ventral root, these come together to form a mixed nerve. All right, they will come together, and we'll see it very, very much in the very next slide. All right, so sensory, as we've said, unipolar, goes into the dorsal root ganglion. Interneuron, a multipolar neuron, receives those inputs, all right, and those inputs will act on motor neurons, all right, and carry out information then as the CNS affects a change on the effector organs, all right, via another multipolar neuron, the motor neuron itself. White matter is axons, and gray matter is regions of cell bodies, all right? And so let's go on to this figure, and we'll say a little bit more about those, the gray and the white matter, all right? When you think about a circuit like that, it forms the basis of a reflex, all right? And reflexes can be defined as simple actions that mediate a direct response to a stimulus coordinated within the central nervous system without input of higher brain centers, all right? So what we're talking about is a simple circuit, a simple information loop that begins with a stimulus. And we can look at a very simple one here, the little patellar reflex here, right? You wrap on the patellar ligament with this little, hammer. It stretches that ligament, which stretches little sensory receptors here in your quadriceps muscle. All right, that carries sensory information in, and here it goes into the mixed nerve. There's your mixed nerve, the sensory and motor traveling together. All right, there's your cell body in the dorsal root ganglion. In it goes into the central nervous system, and it synapses without an interneuron. Hence, this is a monosynaptic reflex arc. All right, just one synapse here in the central nervous system. All right, one synapse, and there's your motor neuron carrying information out, and it stimulates contraction of the effector, in this case your quadriceps muscle, and you kick your muscle, you kick your leg up, right? And so you've got a very simple, a monosynaptic reflex arc here. All right, this is the very fastest kind of reflex arc. Okay, and when you think about it, I think I remember jumping off the table one day for you guys to show you that's the reflex arc that's at play here. All right, monosynaptic reflex arc. You don't want a delay there, do you? All right, you want that to be really fast and be responding to the stimulus that's coming in. All right. On the other side of the figure, you have a polysynaptic reflex arc, like what's called the withdrawal response. All right, if you touch something hot, all right, and you pull your hand away, again, we've got stimulus here, all right, carrying information into the central nervous system, right, our mixed nerve, dorsal root ganglion with its cell body, in we go, and now we have a synapse with an interneuron, all right, and that in turn synapses with the motor neuron down here in the ventral horn, all right, and that carries information out to the effector organ. So this is a polysynaptic, all right, reflex arc here because we have an interneuron, all right. Now, let's say I'm walking along and I step on a piece of glass, all right. I don't want to have to think about what goes on next, or I step on a tack, okay? What do I want to see happen? I want to withdraw this leg, and I want this leg to extend so that it holds me up, right? So here we have now a little more complicated system with a little bit less anatomy to look at, but you get the idea here. So here's our stimulus down here at the bottom. Stepping on the tack, there goes our sensory input. And that sensory fiber, there's its nucleus in the dorsal root ganglion, comes in and it branches, all right? And those branches act on the same side of the body as the stimulus. We call that the ipsilateral side, all right? And they're also going to act to affect changes on the contralateral side, on the opposite side, all right? So what do we want happening on the ipsilateral side? We want to contract the flexor or the extensor? The flexor. So here we go. The branches then act on a interneuron, which is an excitatory interneuron. It triggers an impulse in this motor neuron, hence the flexor is stimulated, right? It also acts on an interneuron that has inhibitory action, meaning it suppresses the impulse being carried in this nerve cell. Hence, no impulse is carried. And what do you do to relax a muscle, skeletal muscle? You don't say anything, and that's what's happening here. So you have inhibition by that interneuron. You have inhibition of that motor neuron, so it will not fire, and therefore the extensor is relaxed, all right? Now, one of those branches synapses with an interneuron that goes across to the other side, all right? So we're going now to the contralateral side, and what's happening here? We've got an excitatory impulse, all right, acting on a motor neuron to stimulate the extensor muscle, and we've got an inhibitory action on the motor neuron so that we do not contract the flexor, all right, and hence it remains relaxed, all right. So there you have a reflex arc that's even more complicated. It's working on both sides of the body, all right, working across the spinal cord, okay. So there you see some basic circuits, all right. Why do we have these circuits? So that you don't have to think about stuff, right? You touch something hot, you don't have to wait for smoke and something's burning and maybe, oh, that's my finger, maybe I should pull it away, right? You don't have to get to that point, all right? Now, when I touch something hot and I pull my hand away, am I aware of it? Do I become aware of it? And can I then say, you know what? I don't want to do that again, right? So now all of a sudden voluntary is going to take over. Well, the white matter that you see here, all right, the white matter is going to carry information up axons, myelinated axons, carrying information up to my cerebrum, telling me, by the way, you just put your hand on the hot plate, all right? And in turn, there's going to be information going down, again, axons in that white matter of the spinal cord, taking information down and acting on the appropriate motor neuron to say, and by the way, you've just pulled your hand away, and let's keep it there. Let's keep that flexor engaged. All right, so now voluntary takes over from reflexive. Okay? There was a hand, Emily. Well, you know what? You know that. You know what you do, right? What do? You try to take the load off right? So you go down, right, by going down to the floor and catching yourself, right? You're taking the load off of both, aren't you? So you can do that, right? Okay. All right. Okay. So let's go back to the cellular level, all right, and let's consider the cellular components of the nervous system beyond just the functional unit, that is the neuron, all right? Neurons make up 10% of the cells, all right, even though they're the ones we talk about most of the time. Neurons only make up 10% of the cells of the nervous system, all right? The other 90% are glial cells, also called neuroglia, all right? These cells are mitotic, and very commonly when people have brain tumors, all right, they are gliomas, and many of you have heard of that term, gliomas, all right? They are cells, or they are cancers that emerge from these neuroglial cells, and it stands to reason. These are cells that are capable of mitotic activity, whereas nerve cells are not, all right? That doesn't mean that nerve cells cannot become transformed, but more commonly it's glial cells that do, all right? And these cells are very important in the development of the nervous system, in guiding the formation of the various architectural connections within the nervous system. They're going to provide support and nutrition and protection. Those of you that have been in the anatomy lab, did you handle any fresh brain tissue? No, it was all preserved. So it was all like a Nerf brain, or it was like a rubber ball. If you dropped it, it would bounce. All right, but what's the real consistency of brain tissue? It's like Jell-O that has been left out for a few hours, right? It's liquid, isn't it? If you held a brain in your hand, you could literally pour it back and forth from one hand to the other. That's a very different tissue in its texture than anything you've looked at, right? You hold a piece of liver in your hands, it's got body to it because it's got connective tissue holding, right? Mesodermally derived connective tissue, all right? What is the brain derived from? Ectoderm. There is no mesodermal component there, and so there isn't any connective tissue, and it has this very liquid sort of consistency, but it still requires some structural support. So this is where your glial cells come in. You've got no connective tissue there, but you have to have supporting tissues. So let's take a look at the glial cells. All right, 90% of the neuroglia are these astrocytes, these little star-shaped cells. And you see one here in this little sketch. All right, these cells are important in forming the blood-brain barrier. So they wrap around the capillaries. They transfer nutrients directly to the nerve cells. So the nerve cells are not getting their nutrition by diffusion across the capillaries, through the interstitial fluid, and into the cell. All of the cells Instead, in the central nervous system, these cells are being fed. They're being hand-fed, all right? And that barrier then between the blood circulation and the nerve cells themselves is called the blood-brain barrier, all right? And it is a product then of these astrocytes, all right, that are screening what nutrients get to those cells, what things don't get to those cells that could be damaging to those cells. It becomes a problem sometimes because if you're trying to treat some kind of problem and you're trying to use a drug therapy and the astrocyte said, I'm not delivering that to the nerve cells, but there's cancer there, I'd like you to do that. And they are filtering out those compounds, all right, and preventing those compounds from accessing the tissues. All right, as I said, these neuroglial cells also help in the development of the brain, and these astrocytes secrete what are called nerve growth factors, all right, and these nerve growth factors serve as guiding chemical signals, all right, to the development and the structural arrangement, the architecture of the brain, all right. Second type of cells are called ependymal cells, and these are cells that line the spaces of the brain, all right, and they are responsible for producing the cerebrospinal fluid, all right. So they line the spaces. Here you have in the brain the ventricles of the brain, the ependymal canal that goes down the middle of the spinal cord, all right. And together with capillaries, they form the choroid plexus. And their job is to produce cerebrospinal fluid, which is an extract of the blood plasma, all right, and it is then used to fill that space. And of course, that space fills those cavities. It also surrounds the brain itself and gives you this fluid cushion around the brain. Edie? Yeah. Yeah. So it's filling these few spaces. It's also protecting the brain on the outside, giving you this liquid cushion. All right, on the outside. It's also as a result of respiration, the pH of the cerebrospinal fluid can change. All right, because it reflects what's happening in the blood plasma, and that in turn is where your nerve cells that control, say, your breathing rate, for example, are detecting CO2 levels and pH levels. So it becomes, in a sense, a communicating medium as well for the condition of the body fluids, all right, in terms of their pH, all right, and CO2 levels, all right. And they are ciliated cells. They're cells that are going to beat and move this fluid, so the fluid is not stagnating, all right. And again, that contributes to that idea that I just said in terms of monitoring the condition of that fluid. All right. Microglial cells are the macrophages of the brain. All right. These are akin to the fixed macrophages that we've talked about in the various tissues, the dust cells in the lungs, the Kupffer cells in the liver, but they are not monocyte derived. All right. They are their own special version. Remember, monocyte derived cells are mesodermal. Right. They're mesenchyme derived. All right. These are cells that are derived from ectodermal tissue. But their job is to clean things up. So when there's damage, all right, when there's nerve cells that have been damaged, these are the cells that are going to remove that debris and those dead cells. And then finally, there are the oligodendrocytes. And I showed you earlier that myelin sheath, that insulating set of wrappings at intervals along that nerve cell, yes, that increase conduction rates. All right, there's a few things that the myelin sheath does. All right, it increases conduction rates, and it also insulates one nerve cell from its neighbor, and in so doing, it reduces crosstalk. All right, and that's the term we use, and I think you can immediately appreciate what that term means. If you have one nerve cell sending an impulse and it's sitting immediately adjacent to a neighboring nerve cell, they may influence one another. So by insulating them, you prevent crosstalk. All right, and again, of course, it also increases the rate of impulse conduction. So this is an interesting architecture because oligodendrocytes, as you can see here, can myelate numerous axons at the same time. So this is one oligodendrocyte, and it's myelinating this one, and it's myelinating a neighbor here, and another one there. Whereas in the peripheral nervous system, you don't have oligodendrocytes. Instead, you have neurolemmocytes, or Schwann cells, and these wrap around a single axon, not extending processes out to wrap around many. All right, and we'll get to those again in just a sec. All right, so we've just looked at the four types of cells that are found in the central nervous system. In addition, the four types of glial cells in the central nervous system. All right, in the periphery, you also need something holding nerve cells together and mediating the nutritional requirements of these cells and so forth. All right, and so we have two types of cells. Satellite cells, again, equivalent to the astrocytes, so they're the maintenance cells. They're the cells that are looking out for, that are filtering and making sure that the nutritional requirements of the nerve cell bodies are being met, okay, of the nerve cell bodies. And of course, you're going to find these in the ganglia then, right? Because that's where the cell bodies are in the periphery, okay? And then the neurolemmocytes, also known as Schwann cells, are going to form this wrapping, all right? And you see it here. This is a neurolemmocyte or a Schwann cell, and it begins to literally wrap itself around until it produces many, many layers of plasma membrane, all right? And it's just layer upon layer of phospholipid bilayer, all right, which is serving as this insulating component. And these are, 1/10 of a millimeter insulation and then a little gap, and 1/10 of a millimeter of insulation and a little gap. All right, and that allows for conduction along the nerve cell to occur. If you have an unmyelinated axon, every point along the axon has to undergo a nerve impulse as that impulse is conducted down the length of the nerve cell. If you have a myelinated axon, all right, we put this wrapping of these neurolemmocytes or Schwann cells, it means that the impulse jumps here and then here and then here. And as I said, 0.5 meters per second, 120 meters per second, all right? This jumping kind of conduction is called saltatory, all right? Jumping in Spanish is un salto, right? A jump, all right? So saltatory conduction, all right, is a function of having this wrapping of neurolemmocytes around the axon, or what is referred to as a myelin sheath, all right? And this is characteristic of all the nerve cells in the periphery, right? All the nerve cells in the periphery to allow this very fast mode of conduction. All right. Regeneration. What do you know already? We said that if you damage the nerve cell body, that's it, right? Somebody has a spinal injury, they have a compression of the spinal cord, it crushes those nerve cell bodies in the spinal cord. They never regain motor function, right, of those tissues that are below that point of injury, all right? You can damage nerve cells, though, as they extend out into the periphery. That is, you can damage the axon and still get regeneration. The further out toward the periphery, the more distal the injury, the better chance of healing, because regrowth requires only a shorter distance, all right? Damage way up here for a nerve that goes all the way down to here to allow me to move my fingers, all right, means regrowth down a very long distance, and the chances of that occurring are diminished, all right? But the basic idea is if you sever then, and how many of you have ever, you got a deep wound and you lost sensation, all right, and it took how long for you to regain sensation? Days, weeks, months? Months, maybe 6 months, maybe sometimes years. Yeah, all right, because it's got a long way to grow. They say that nerves, the axon regrows about the rate of your fingernails growing. I don't know if that's a good estimate, but I think the inference is it's really slow going. And what happens is you've severed the axon, all right? The proximal portion seals off and swells up, and so here you see the proximal portion, and the distal portion is reabsorbed, okay? And the neurolemmocytes then begin to form a regeneration tube. And this, so you're using the neurolemmocytes that were there. You're using the myelin sheath that was already there to form a tract for regrowth. That means that when you have that injury, you better realign the cut end with its former component, right? If you don't realign them, the chances of regrowth are zero. All right, so you try your best to realign them. And when you do that, then the neurolemmocytes form a corridor of sorts, all right, to allow the axon then to begin to regenerate and grow out and eventually produce its synaptic terminals and reinnervate, all right, plug into the tissue once again, so reinnervate the tissue, all right. All right, we'll do this slide and we'll finish here, you guys. When we look at nerves in the periphery, a nerve is a bundle of neurons, right? And we said that in the central nervous system there is no connective tissue, but in the periphery there is, all right? And so in the peripheral nervous system, the nerve itself is the bundle of axons, if you will, is supported by areolar connective tissue and the neurolemmocytes, all right, and capillaries that are supplying oxygen to those cells, all right? That is bundled up into fascicles by, again, a dense irregular connective tissue sheath that gives you, that's referred to as the Perineureum, all right? And then all those fascicles are bound together with a thick superficial dense irregular connective tissue that ensheaths the entire nerve, all right? So we've got the Endoneurium at the level of the axon, You've got the Perineureum at the level of bundles of axons, all right, and you've got the Epineurium ensheathing the whole thing, all right, to give you the nerve itself, all right, and we'll stop there.